So chapter 15, so the importance of energy changes and the electron transfer in metabolism. So the first few sections of the chapter 15 basically discuss about this the reaction rate, how to perform, how to calculate the reaction rate. All right. So for standard mathematical representation of reaction rate, so for example, this decomposition. All right. So from N2O5 decomposed to these two products, and then for the rate of the reaction, and the rate of reaction is proportional to the concentration. Okay, therefore the concentration of the N2O5 is denoted as this, and then the rate would be like this, and this is the rate constant. So you learn all this in the general chemistry. Okay, so for this reaction, the reaction rate is written as this, right? K is a rate constant multiply the concentration of the reactant. Okay, the reactant. So that's reactant and product. Okay, and so for a reaction from A, that's the reactant, to the product. So and then you write the equation, the rate equal to the rate constant multiply the concentration of the reactant, and then to the power of the N, that's the order of the reaction, which must be determined, right, experimentally. All right, that's it. So that's the order of the reaction. Okay, and for the equation like this, A plus B, the reactant, you have two reactants, A plus B to the product, then the rate equation would be like this. The rate constant multiply the concentration of A to the power of N multiply to the concentration of B to the power of M, right? Of course, these are the reaction order determined experimentally. Okay, so that's the equation. Okay, and then for this, it's just the same. That's oxidation of ethanol, and then so the concentration will be this ethanol multiply the oxygen and then multiply the rate constant. Okay, so that's the rate equation. And then for the equilibrium, so the equilibrium reaction, basically that means the chemical reaction do not go to the completion, right? So basically it's an incomplete reaction. That's the equilibrium reactions. Okay, so for the equilibrium, then you have the reversible. It's a reversible reaction, and then you can see a symbol like this. A process can occur in both directions. Okay, and the rate of the forward process in a reversible reaction is exactly balanced by the rate of the reversed process. Okay, it's a dynamic equilibrium, and it's a reversible reaction. And yes, Hadel.
Professor, why don't you take the products? We're just gonna do the rate reaction for the reactants, not the products, right?
No, that's if it's only one direction here. That's one direction. Did you see the arrow is one way?
Yeah.
Then the rate will be only depend on the concentration, concentration of the reactant. Okay, and then if it's reversible, then that's what are we going to discuss now for the reversible. So for example, like this, the reversible reaction. Then in the beginning, okay, the reactant this direction to the right. So this will be very fast, okay? And then slowly from the product go reverse reaction, slowly increase. And in the end, they will reach the equilibrium. So that means the forward reaction equal to the reverse reaction. That's when you reach the equilibrium. So that means the reactant depletion is equal to the rate of the product depletion. Okay, so the rate of forward and reverse reaction are equal. And then you can introduce the idea from the beginning that I just showed you. For the forward reaction, right? Nitrogen plus hydrogen to the ammonia here. So the forward reaction will be like this: K rate F forward equal to Kf multiply the concentration of nitrogen to the power of n and the hydrogen to the power of m. And then rate R, that's reverse from the ammonia back to the nitrogen and the hydrogen. So that'll be the concentration of these. And since the equilibrium, the rate of F will equal to rate of R, right? Like this. Okay, and then K equilibrium equal to Kf over Kr, because this item, this item equal to this item. Therefore, you can move these two to the other side. So see, originally Kf multiply N2H2, right? Kf N2H2 equal to Kr and H3. And now you just move Kr to here, move N2H2 to here. So that give you K equilibrium. So that'll be Kf over Kr equal to this. So that's the rate of the equilibrium.
Professor, but
Yeah.
over here, can you go back 1 minute? So it says over here that Kf, the rate of F, which is the Kf, is equal to N2N H2M, right? But over here
Right.
in the equation, the picture of the equation, it says NH3P. Oh,
Right.
but it's not equal, like it's not like that. It doesn't look like that. Why is that?
Yeah. This one should be equal to this. All right. And that's why if you look at this Kf, and then because when they reach the equilibrium, the rate F should equal to rate R. All right. So that means Kf N2H2 equal to Kr NH3. So that'll be, if you move these two items to here, so that'll be Kf multiply N2H2, and move this item to here, that'll be equal to Kr multiply NH3. All right, so that's how they derive this equation. This, if you multiply this, will equal to this multiply this. So now you just move Kr to here and move N2H2 to here.
Okay.
So that you can get this ratio for this.
I see.
No.
So basically, we're cross multiplying it to equal.
Yes.
Okay. Okay.
Yes. Okay, that's very good. So that's how we get this K equilibrium equal to Kf over Kr and equal to this NH3 to the power P over the N2 power of N multiply H2 multiply of M. Okay, so that'll be the KEQ, that'll be constant at a constant temperature. So now if we have this the equation, now be AA plus BB to CC plus DD, right? And AB, they are the reactant, CD are product. So the K equilibrium now be equal to these two product multiply the product of this product and then the product of this reactant over the reactant. Okay, the C to the power of C multiply D to the power of D over A to the A, B to the B. Okay, so that's reaction constant for the equilibrium, the equilibrium constant. Right, so that's basically all these are just similar. So this very similar reaction. Where, in which one, Adele?
So, Professor, what if we're adding energy to break bonds instead of forming bonds, right? So, for example, if we're adding like heat, right? Is the equilibrium gonna transfer to the left or the
No, that would be different if you add the energy
Mm-hmm.
to make the reaction happen.
Yeah. Hello?
Yes, if you make the energy, yes, if you added the heat to the reaction, and so then it's not a spontaneous reaction. So you add the heat, and of course that will be taken into account when you do the rate constant. That will be considered as well. And then to interpret the equilibrium constant, okay. And then if the rate constant is between the 10 to the minus 3 to the 10 to the 3, so that means the equilibrium mixture contains significant concentration of both reactant and of the products. If it's smaller than 10 to the minus 3, all right, so that means the most the reactant present. Okay, so Mona, what's your question?
Good afternoon, sir. I thought there is an exam at 4 o'clock today.
Yes, and now I just decide to push back a little bit, okay? To push back about until 4:20.
Oh,
Okay.
okay, sir. Sorry, I didn't know that, so I missed a little bit of the class.
It's okay, don't worry. Yeah, it's also a miscommunication. I should have told you Friday, but it's okay. All right.
Okay, thank
Good. And then the standard free energy. So for the standard free energy, assume a concentration of 1 molar and pH 0 for biochemical reaction. So we define a different standard state for the concentration of the hydrogen. So that will be the 10 to the minus 7, which is pH 7. Okay. So that's for the biochemical reaction. And for the pure chemical reaction, then they define as a pH 0. Okay. For biochemical, it's about the pH 7. And here, just to show the biochemical connection through the thermodynamic, and we know the hydrogen in the sun, then that can be absorbed, and then through those plant, and then they perform the photosynthesis, and then to produce the glucose, right? So and also other sugar in the plant, and then also the production of ATP basically is come from the oxidation of glucose. They will release the hydrogen and also electron, and then through those transfer, then you make the ATP, all right? And then this ATP can be used to synthesize these protein and other like a lipid, protein. Those are macromolecule. Okay, so this is the connection between the thermodynamic, and those are biochemical. And now the metabolism, basically that's chemical reaction of biomolecule. And then we have catabolism, which is the breakdown of large molecules, just like you break down the glucose. And anabolism, the synthesis of large molecule from smaller one, right? And for breakdown, basically it's oxidation process, right? Because you oxidate, for example, oxidated glucose, and then those coenzyme, they will be reduced. So that means you transfer the electron and hydrogen, the electron to those coenzyme, and coenzyme can carry those electron, hydrogen, then to the mitochondria. Then that's the place. Then we synthesize the ATP. All right. Then they can transfer the electron and then to make the ATP, and that's the catabolism. And it's a breakdown of the large molecule with oxidation. All right. And then synthesis of large molecule from the smaller one, that's anabolism. All right, which is a reductive process and require the energy. So and then metabolism is the sum total of the chemical reaction of biomolecule in an organism, and from here, so it doesn't matter whether it's a fat, polysaccharide, protein, right? And then they have to perform the oxidation, the catabolism, all right? Catabolism, and then oxidation. So they break down this into the smaller molecule, polysaccharide to the glucose, another monosaccharide, and fat, you have fatty acid and glycerol. Protein is amino acid. And then they continue to break down to oxidize these molecules, then become even smaller molecule, all right? And then those molecules either be excreted or they can go back for anabolism, which is here, right? And that will be the reduction reaction here. And then to make the large molecule. So that's the process. And then it's a metabolism. And then during this process that I mentioned, oxidation and the reduction, okay? So the oxidation, reduction, oxidation, loss of electron, all right? Loss of electron. And then they are the reducing agent because they will reduce the other one that is subject to the reduction, and themselves subject to the oxidation. And the reduction, the gain of the electron, okay? That's electron, and the gain of electron, they are oxidizing agent. And the most reduced form of carbon is arcane here, and the most oxidized carbon is here, carbon dioxide, all right? And from here, okay, most reduced, that's because they are going to be oxidized, all right? Because they're going to lose the electron. And here, they're going to gain the electron, okay? So they will be reduced, all right? So here is most reduced, and here is the most oxidized. So they will reduce this direction, they will oxidize this direction, all right? So oxidation, basically, loss of electron, that's oxidation, loss of hydrogen atom, gain of an oxygen atom, all right? So loss of the electron, loss of the hydrogen, and gain the oxygen, all right? So they are oxidized, and right, loss of electron, loss of hydrogen, gain oxygen atom, reduction, gain electron, gain hydrogen, and loss of oxygen. So from here, they will gain the electron, gain the hydrogen, this direction, right? So they'll be reduced, and here's be oxidized. Okay, so in here, so magnesium lose the electron. Magnesium is oxidized, all right? Chlorine gain the electron, is reduced. So this is the oxygen oxidizing agent, right? This is reducing agent, okay? So that's oxidation and reduction. So when the reaction is at equilibrium, delta, the free energy change is 0. And then, so basically that's a summary of this catabolism, large molecule broken down to the smaller one. Anabolism, small molecule react to give the large one, okay? Then the coenzyme. The coenzyme is important, okay? Because, for example, the nicotinamide adenine dinucleotide NAD+, very important coenzyme. Later on, when you learn the glycolysis, you will learn how important the NAD+ is, right? Because they are the oxidizing agent, and they are going to be reduced, and they cause the glucose oxidation during the glycolysis, right? During the glycolysis. It's very important coenzyme, glycolysis and citric acid cycle, and then later on in the ETC, electron transfer chain, those are coenzyme will then be, because they will be reduced. Then later on, when they receive the hydrogen and the electron, then they will be oxidized. At that time, they release the hydrogen and electron, and then to produce the ATP. Okay. So and then the NAD plus is a 2-electron oxidizing agent. All right. So 2-electron oxidizing agent. So it's here, then it will be reduced. Okay, it will be reduced. So this is reduced form. All right, during the glycolysis and the citric acid cycle, it's a, and then when they are reduced, then later on in the mitochondria, the ETC, these electron, okay, will be transferred, be released, they will be oxidized, so that those electron goes to the ETC to produce the ATP. Okay. And then another important coenzyme is called flavin adenine dinucleotide, FAD. It's also the oxidizing agent, also used to oxidize the glucose during the citric acid cycle. So this is the oxidized form, and then this is reduced form. All right. So during the citric acid cycle, so they receive these 2 hydrogen and 2 electron, so they become reduced form. And here is the place that they receive. They receive the hydrogen here, and here. So that's where you see these 2 hydrogen. Okay. And this is just the structure of this riboflavin, and then the flavin mononucleotide, FMN, and then also the FAD, the dinucleotide. So the ATP phosphorylation, okay, so the phosphorylation of ADP to ATP require the energy, and then if you hydrolyze, the ATP will release the energy. Okay, that's because this high energy bond, right, between alpha and beta phosphate and beta and gamma phosphate. These are the high energy bond, and the high energy bond refer to the fact that they require or release convenient amount of energy depending on the direction of reaction. If you're going to phosphorylate it, then you're going to supply the energy. If you're going to dephosphorylate, then you're going to release the energy. Okay, so that's why we say these are the high energy bond. Okay. And the ATP is less stable than the ADP. That's because energy must be expended to put an additional negative charge on ATP. And also the entropy loss when ATP is phosphorylated. So there is a potential loss of resonance hybridization of inorganic phosphate upon the phosphorylation of the ADP to the ATP. Okay. So this is just to show the loss of a resonance stabilizer phosphate ion in production of ATP. So when the ADP, all right, the ADP is phosphorylated to the ATP, there's a loss of the resonance stabilizer phosphate ion. That would result in the decrease in entropy, all right? So that's the production of ATP. And And here's just to show the decrease in electrostatic repulsion on the hydrolysis of the ATP to the ADP, and also hydrolysis ADP to the AMP. And that will lead to a relief of the electrostatic repulsion. Okay. Yeah, here just to show you the you have these nutrient like the glucose and then catabolism. So basically oxidation of the glucose and then break down the glucose to carbon dioxide and the water. In the meantime, they transfer the energy, right, to produce the ATP. And then the ATP can dephosphorylate and then release the energy, and the energy help those precursor small molecule to perform anabolism and then to form the large molecule. So the coenzyme A in activation of metabolic pathway, the activation that means the formation of more reactive substance. Okay, so like this A plus coenzyme, and then become this complex, and it's A linked to the coenzyme, that's the activation step, and then the A coenzyme plus B, and then to the AB, and then release the coenzyme, and then it's asynchronous reaction. And then it's the spontaneous because the energy change is negative. Okay. And the coenzyme contain the unit of two capital acyl amine and pantothenic acid and 3' 5' ADP. Okay. So that's the coenzyme A, and they can help to form this more reactive substance. Okay. And this is to show the electron transfer and the ATP production. As I mentioned, the catabolism, you break down the big molecule like glucose, all right? Glucose, fat, all right, and protein, and then it's through the oxidation, all right? And then they will enter the citric acid cycle, and during this citric acid cycle, as you can see, this NAD plus FAD, these two will be reduced, and then after the glycolysis, right, the pyruvate acid, that's the end product of the glycolysis. They enter the citric acid cycle, right, and then they will continue to be oxidized, and then in the meantime, these two coenzyme will be reduced, all right? So they transfer the hydrogen and electron to the NAD plus and the FAD, so you become the NADH and the FADH2. And then these coenzyme, the reduced coenzyme, will enter the ETC, electron transfer chain, all right? And during that ETC, then they can transfer the energy and to transfer the hydrogen and electron so that they can phosphorylate ADP to make the energy ATP here, all right? And this process, it's oxidative phosphorylation. Okay, that's catabolism. And then if you're going to perform anabolism, all right, so that means those reduced form will be oxidized, and then small molecule will be reduced, and then to form the large molecule. Okay, so that's basically the chapter 15. Any question?